Hello, everyone, and welcome to today's webinar titled Characterization of Residual Impurities in Gene Therapy Drug Products, hosted by Biopharma Webinars and presented by Jessica Townsend, Associate Director of Analytical CMC Lead at Intellia Therapeutics. Jessica, hello there. Welcome. Thank you for joining us today.
Hi, Marcus. Thanks for having me.
It's a pleasure. And following Jessica, we also have Eric Bishop, Vice President of Research and Development at Cygnus Technologies. Eric, hello there. Welcome, and thank you for joining us as well.
Thank you, Marcus. Thanks for having me.
Yeah, it's a pleasure. So I'll be today's moderator. My name is Marcus, and before we begin, I'd just like to inform our viewers that there will be a live Q&A session after both presentations before the end of the webinar. So audience members, please feel free to send in your questions at any time via the questions tab located directly below your webinar screen, and we will go through these during our Q&A session coming up after Eric's presentation. And please also direct in your questions to the relevant presenter. So if it's a question for Jessica, please mention so in your question, or the same for Eric as well. Okay, now please allow me to introduce our first presenter. We have Jessica Townsend, who is Associate Director of Analytical Development at Intellia Therapeutics. And Ms. Townsend received her bachelor's in biology from Minnesota State University and her master's in molecular and cellular pharmacology from the University of Wisconsin-Madison. Following graduation, she spent 10 years developing molecular diagnostics and infectious diseases using PCR techniques, and was instrumental in several successful FDA F10 submissions prior to making the transition to gene therapy. Ms. Townsend joined Bluebird Bio from 2018 to 2022. Most notably oversaw the residual impurities methods utilized in testing lentiviral, vector, and ex vivo gene therapy drug products, authorizing impurity sections from 3 BLAs in her time with Bluebird. And recently, Ms. Townsend joined Intellia Therapeutics as the ex vivo CMC analytical lead, representing analytical technologies within the ex vivo CMC teams across Intellia. And with that, I'd now like to hand over to our first presenter today. So welcome, Jessica.
Thank you, Marcus. Okay, so today I'll be talking about characterization of impurity reduction in ex vivo gene therapy drug products, as Marcus stated. My name is Jessica Townsend, and I am Associate Director at Intellia. And let's get this show on the road, shall we? Okay, so for today's agenda, we'll start by talking about an overview of the ex vivo gene therapy manufacturing approach. Then we'll move into what exactly are residual impurities and why do we care. Following that, we'll talk about CQA assessment and process characterization of impurity reduction. We'll go on to talk, actually the majority of my talk will really be on regulatory guidances on impurities and how you can use those guidances to your advantage. And then I'll just give a quick summary of my talk. So we'll start with the overview of the ex vivo gene therapy manufacturing approach. So this is a schematic of a very generic ex vivo gene therapy manufacturing approach showing a CAR T example. Cells are removed from either the patient for an autologous therapy or a donor for an allogeneic therapy. They're shipped to your manufacturing organization where manufacturing is performed via ex vivo selection, activation, editing, insertion of the gene of interest, and expansion. So you'll have a selection for your T cells, and then there's an activation and transduction process wherein your viral vector, in this example anyway, your viral vector is added to the cells. And after that, there's a wash, an expansion, and I don't show it here, but generally a cryopreservation following prior to infusion into the patients. And if we go on to the next slide, we'll see just showing here some of the areas where you might be seeing an introduction of residual impurities into your process. For example, your labeling reagents during your wash and selection, your vector and transduction enhancers following your activation transduction. And then in your final product, you'll also see unedited cells, non-target cells, and non-viable cells, which are also considered impurities in your process. So what exactly are residual impurities and why do we care, right? So impurities, as defined by the USP, an impurity is any component of a drug substance that is not the chemical entity defined as the drug substance. And in addition, for a drug product, any component that is not a formulation ingredient. So basically anything in your product that isn't your actual product, right? So drug product impurities are generally divided into two separate categories. You have your product-related impurities, which originate from the cells themselves. And so this is where you find like your dead cells or your non-target cells or your untransduced cells. Those are all considered product-related impurities. But what I'm really going to focus on for this talk is the process-related impurities, which arise from the manufacturing process. So that includes your raw materials used in the manufacturing process. For example, you might include human serum albumin. That's not necessarily something that you're trying to infuse into your patient, but it is an impurity in your process. Or things like cytokines that you might add in. Those are your raw materials that are impurities in your final product. And then also your vector-related impurities are a big component of your process-related impurities arising from the manufacturing process. Here, I'm really going to focus more on viral vectors, although I know that there are ever more vectors being developed within the industry. But in this instance, we'll talk about the viral vectors. So moving on, why do we care about residual impurities? Well, I mean, for starters, consistent impurity reduction really helps to establish that your manufacturing process is in control. Additionally, impurities can affect the efficacy and stability of your product. For example, proteases from your host cell proteins might really affect your stability if you aren't able to reduce them to nice low levels. But most importantly, really, the control of the level of impurities in a drug product is necessary in order to ensure safety. So there are multiple safety concerns regarding impurities. For starters, toxicity. Reagents used in your manufacturing process might be toxic depending on the levels that they are present in your final drug product. And these can be assessed through literature searches. Immune responses are also something to be concerned about when you're considering your impurities within your product. As I mentioned before, host cell proteins and also DNA can cause immune responses. And finally, oncogenicity caused by insertional mutagenesis is always a big concern to the regulatory agencies and, of course, to us. Residual vector is a concern in this regard, residual DNA, residual competent viruses, should you have those in your product as well. So next, we'll talk a little bit about CQA assessment and process characterization of impurity reduction. So critical quality attributes, or CQAs. Per ICH Q8, CQA is a physical, chemical, biological, or microbiological property or characteristic that should be within an appropriate limit, range, or distribution to ensure the desired product quality. So basically, any quality of your drug product that you need to have within an appropriate range or kept below an appropriate limit is considered a critical quality attribute, right? So CQA identification supports process development. So you might want to look at you might want to ensure that you're maintaining quality while making tweaks to the process. And so ensuring that your CQAs are within a certain range during process development is very important. Determination of your control strategy is important for another area where CQA determination will be important. Knowing where you might want to put your in-process controls, what you might want to control on release, what you might want to monitor throughout your process, all of that will come into the CQA assessment. If you're performing comparability, you want to make sure that your CQAs are maintaining their levels within a certain range. And then, of course, the safety assessment of your product. So when you are submitting to the regulatory authorities, safety assessment is especially important, and your impurity CQAs will come into account in that regard. So when you're looking at your quality attributes, you really want to be able to separate them in early in the process into your critical quality attributes, those that you know to be critical before you even begin, your potential critical quality attributes, your PCQAs, and then, of course, your non-CQAs. So your PCQAs can actually become CQAs or non-CQAs based on your knowledge gained during development or characterization. But knowing that they are potentially a CQA will be helpful during your process development. You can determine what you have as far as CQAs, PCQAs, and non-CQAs early on in your process based on the predicted severity of the impact to patient safety or efficacy, and the uncertainty or certainty in knowledge of this impact. So this slide shows kind of a generic risk assessment for potential CQAs, right? So on the left, we show a table for severity of impact where you can define your quality attribute as either low, medium, or high severity to your product, to either the safety or to the efficacy, and really defining what you mean by low, medium, and high for both safety and efficacy is important here. In order to use this to define what are your CQAs, your PCQAs, and your non-CQAs. And another thing to point out is even though there are definitions for low, medium, and high impact to both safety and efficacy, you're going to want to err on the side of caution here. So if you're seeing high impact to safety, but maybe only a medium impact to efficacy, you'd consider that a high severity. On the right side, I have a table for uncertainty. So you can look at low, medium, and high uncertainty. And there are several different areas that you might want to look at as far as where you might gain your knowledge for your impurities, right? Prior knowledge based on characterization of other products or published literature, or just in your past experience, prior knowledge can be used. But also the in vitro studies, animal studies, and clinical studies, either within your organization, prior organizations you've worked in, or really the literature is a really great place to go to determine what the uncertainty level is for an impurity. And when you combine the severity and the uncertainty, you can use this to really determine whether you have an absolute CQA on your hands, a potential CQA, or a non-CQA, as shown in the table below. So here's just a pretty easy example of a risk assessment of a potential CQA. So in this example, cytokines are considered potential CQAs. In this example, the cytokines are used for pre-stimulation of the cells prior to transduction. They enter the process at levels greater than the permissible daily exposure. And I'll talk about what the permissible daily exposure, or PDE, is and how you can define this a little bit later on in this talk. However, in our case, any instance where an impurity was introduced at higher than this permissible daily exposure, the severity was rated high due to potential for the adverse events. So the severity here is rated high. However, cytokines, they're well known. They've been studied in clinical trials. Their impact is pretty well understood. So the uncertainty of cytokines was ranked as low. However, anytime we have a severity ranked as high in our process, we consider that a CQA. So you can see on the table, it's both a high severity, low uncertainty, and so we choose this is a CQA because it is a high severity. Moving on, we'll talk a little bit about process characterization of impurity reduction. So during process characterization, it's really important to test multiple points in your manufacturing process to understand the reduction of different impurities with special attention to your CQAs, but even your PCQAs, well, especially your PCQAs too, because you're really trying to understand and learn how they affect your product. But even your non-CQAs, you're going to want to do a decent amount of characterization just to understand the levels that they are at within your process. So one thing I'd like to point out here is that you're going to want to look at drug product impurity reduction throughout your drug product process, but you'll also want to look at it throughout your drug substance process. So for example, your vector, you'll want to look at the different impurities within your vector and how those are reduced throughout your process in order to really understand your process better and to understand how to maybe improve your process or how to really justify the levels of impurities that you have. So you might you might calculate the total amount entering your process, and then you might want to sample at, say, the point that the impurity is introduced, and during each of your reduction steps as well. So if you have multiple washes during your process, I would encourage you to test for impurities in those washes, and to really understand what your impurity reduction is, and then again your final drug product, if possible. It is possible at times that you will find your matrix of your final drug product will interfere with whatever method it is you're using to quantify your impurities. And in that case, it's perfectly fine to make a case for testing in maybe your final wash or your final drug substance prior to formulation, and using that as your final concentration. One way to really characterize the reduction of any impurity is to calculate your log reduction value. And that can be using the calculation below. So your log reduction value, or your LRV, is calculated using a log base 10 of the total amount entering into the process divided by the total amount in the end product. And you actually can calculate this throughout the process. Like, you can calculate your log reduction value from, say, the amount entering the process to sample point 1 to see what kind of reduction you get from that perspective. Or you can do it, and I would encourage you to do this for the amount entering the process all the way down to your final drug product. And you might not always know the total amount entering into the process, so it's perfectly fine to start from, say, sample point 1 and get your log reduction value to characterize this impurity reduction down to your final drug product. The example below shows LRV log base 10 of an impurity that enters into the process at 100 milligrams, divided by, say, the concentration that you've detected in your final drug product was 0.001 mg per mil. And let's say your final drug product is, for simplicity, is 100 mils. So this would be 100 divided by the 0.1 milligrams total, and log base 10 of this is a 3. And so this is something that you can use really to compare your log reduction throughout the process for the different points too in your process, or not in your process, but in your development process. So from your PD batches to your ENG runs to your PPQs, you can do a comparison throughout those to really show that you're maintaining consistency throughout the process from early on in your stages of development, from your phase 1 all the way to phase 3, by showing that your log reduction values are staying pretty consistent throughout. Moving on a bit to the regulatory guidances on impurity. So this is a little bit funky, but so this slide just shows some of the guidances that you can find on residual impurities. ICH Q3A, B, C, D. There's an M7 and MAP 5017 are all really helpful. The one that I'm really going to concentrate on here is ICH Q3C, the impurities in residual solvents, because that really gives us the tools that we need to set our permissible daily exposures. And in gene therapy drug products, what I found is that a lot of our residual impurities aren't specifically called out in these guidances. And so you really need to be able to set your permissible daily exposures based on the literature and using your own calculations. There are residual impurities where the permissible daily exposure is set specifically in the regulatory guidances, especially Q3C and Q3D. They set permissible daily exposures for certain impurities, especially the elemental impurities. But for a lot of the impurities that you might see within your gene therapy products, they're not going to be here. For example, your host cell proteins, they don't list the host cell proteins permissible daily exposure within these guidances. So that's something you kind of have to determine on your own. And I'll talk about that a little bit more further on. So for impurities where clinical data are available, a safety assessment based on the comprehensive toxicology review should be conducted. This is really your first line of defense, your first line of, like, reasoning for how to understand what levels may or may not be safe within your product for a specific impurity. So the permissible daily exposure level can be derived for each impurity from published non-clinical and clinical data. And again, I'm calling out ICH Q3C and Q3D as really the most relevant guidances that I've found for residual impurity characterization. So within Q3C, and actually within Q3D, but I think Q3C goes into a little more detail, they give you this equation for the permissible daily exposure. And this can be used to justify where a safe level of any certain impurity can be found. So within this equation, you need to specify either a NOEL or a LOEL. The NOEL is the no observed effect level, and this is preferred. So finding something in the literature, some sort of article that describes infusion into a patient of your impurity where there was no observed effect level, that's really the preferred route. But if you can't find within the literature something where there was no observed effect, the lowest observed effect level can be used. So that's the LOEL. Weight adjustment, generally the weight adjustment for an adult in these equations is, again, just erring on the side of caution, which is pretty consistently something that you'll see in impurity reduction and in these safety assessments, is that you want to err on the side of caution. So you can use the LOEL, but for the weight, sorry, got a little lost there. But for the weight adjustment, 50 kilograms is kind of erring on the side of caution for an adult. For pediatrics, I think about 20 kilograms is usually pretty standard for erring on the side of caution for children. There are also a bunch of factors that are included in the bottom to ensure, again, that you're taking the most comprehensive and the safest approach to determining your permissible daily exposure. So F1 is a factor to account for extrapolation between species. So if within this article that you are citing for your PDE, it was a mouse study, you can use an F1 factor of 12 for mouse to human. And these are all laid out for you too in Q3C. So that 12 for mouse to human is specifically laid out in Q3C. And they have them for, let's see, there's for dogs, for monkeys, for rats, all of these different factors to account for the different species that you might use. F2 is a factor of 10 to account for variability between individuals. I see this used in pretty much every calculation. I would encourage you to include it in yours as well. F3 is a variable factor to account for toxicity studies of short-term exposure. So if you're looking at a study that's maybe a 3-month study for rodents, for example, or a 2-year study in non-rodents, you would use a 5. And again, these are spelled out in Q3C, which is super helpful for ensuring that you are following the guidances accurately. F4, a factor that can be applied in cases of severe toxicity. And again, they talk about what numbers you might use. I believe it's usually around 10 or 12 for if within the article that you're citing, there was a case of severe toxicity. And then F5 is a variable factor of up to 10 that can be applied if the NOEL was not established. So if you're using the LOEL instead of the NOEL, you're going to want to include that F5 factor. Moving on, here's just an example from the guidances. I think this was in Q3C, but a toxicity study of acetonitrile in mice was conducted, and the no observed effect level, the NOEL, was calculated to be 50.7 mg per kg per day. And so the PDE for acetonitrile in this study can be calculated using that 50.7 mg per kg per day. You'll slot that into the NOEL spot and then use a weight adjustment factor of 50 kilograms, like I spoke about in the previous slide. And then on the bottom of the equation, you have a factor 1 of 12 to account for the extrapolation from mouse to human. Factor 2 is a 10 to account for the differences between individual humans. Again, I use that every time. F3 is a factor of 5 because the duration of the study was only 13 weeks. And again, that's spelled out within the guidances. And then for F4 and F5, those are both counted as a 1 because this was a NOEL and no observed effect level instead of the lowest observed effect level. There was no severe toxicity encountered within the article, and the NOEL was determined. So as you can see at the bottom, the permissible daily exposure for acetonitrile was found to be 4.22 milligrams per day. So this is great. You set your permissible daily exposure, but what exactly do you do with that, right? So the PDE for acetonitrile, 4.22 milligrams per day. In our instances for gene therapy, generally we don't dose day after day after day. And so we are taking we're erring on the side of caution again here by using the permissible daily exposure for something that we consider a permissible single exposure for 4.22 milligrams per dose. So based on the dose volume, you can actually calculate a maximum permissible concentration. So if instead of a total amount of impurity you're curious about, you can actually you can change this to a concentration, which is pretty basic calculation, right? For a dose of 100 milligrams of 100 mils, you would take that 4.22 milligrams and divide it by 100 to give yourself 42.2 micrograms per mil. So now, great, you have a permissible daily exposure or a permissible single exposure, 4.22 milligrams per dose, and a concentration of 42.2 micrograms per mil. There are a couple of different ways to approach determining if the process meets this criteria. So you can calculate the input amount of, in this case, acetonitrile, and if what you calculate going into your process is below your permissible daily exposure, 4.22 milligrams, you're golden. You don't really need to worry too much about this impurity, right? You're already below the permissible daily exposure. You're still going to want to characterize the reduction in a couple of batches to show that you're actually detecting you're detecting the amounts and that you're seeing impurity reduction, and you're going to want to show that in your regulatory filings as well. But overall, you don't have to put together a big risk assessment. You don't have to justify your amounts too much. You've done the work, you've done the calculation, you're below the amount going into your process. If your input amount is greater than your permissible daily exposure, you'll want to test your final drug product for residual acetonitrile with a method sensitive enough to show this reduction below your maximum permissible concentration of 42.2 micrograms per mil. And again, you're going to want to characterize that throughout the process and also calculate your log reduction values. But assuming that your process does a pretty good job of clearing out the acetonitrile, you should have a pretty good story to tell the regulators. If you can't show if your method isn't sensitive enough, you can show reduction through spiking studies as well. In which case you might use a scale down model or a process development model of your process, spike in your impurity at a higher concentration early on in the process, and then test for it throughout the process and show what the log reduction value is and like what you would expect to see as far as how much reduction of your impurity you would have throughout the process. So there's that. You can also show theoretical reduction using a surrogate analyte, and I found that to be helpful in the past if there's something that you can't calculate for. And I'll show an example of that next, actually. Or you could also, last line, your last kind of hope, is performing a risk analysis to support the levels observed. And in that instance, I've always gone to a toxicology consultant to perform a risk analysis. And generally, they'll go through the literature. They'll see what they can find as far as risks associated with whatever impurity it is that you have, and they'll perform a risk assessment to see what they feel is the concern there. I'll try to speed this up a little bit. I am almost done, though. I know I'm running late. So in this slide, we'll talk about showing theoretical reduction using a surrogate analyte. So for example, host cell protein reduction in drug product where your host cell is human-derived can be a bit of an issue. So for example, if you are growing up your vector in HEK cells, which are human-derived cells, and then your vector goes into your drug product, which is also human cells, well, since the drug product and the host cells are both human, cross-reactivity of the HCP ELISA with the drug product doesn't really allow for testing in your drug product matrix, right? Luckily, your HCP is carried over from the vector into the drug product at levels controlled by the vector material specification. So what you can do is you can calculate the maximum amount of host cell proteins that would go into your drug product. You can, based off of your material specification, you can calculate your max vector volume times your going into your drug product times the maximum amount of host cell protein you would allow in entering into your product. So a surrogate analyte with similar physiochemical properties, for example, here, human serum albumin might be used with adequate data on impurity reduction. You can use that to support your reduction of an impurity. In order to do this, to calculate your maximum amount in your final drug product, you put in the maximum input amount of your host cell proteins in the top of the equation and divide that by 10 to the LRV of the human serum albumin that's already been determined empirically. So in this way, you can show maybe that your host cell proteins are below, or maybe not below, your permissible daily exposure, depending on whether or not you've been able to calculate that for host cell proteins. But you can show that they would be reduced to a certain amount within your drug product, and you can show that to the regulatory authorities in your submissions. So HCP ELISAs, due to the inherent variability in biological systems, the amount and composition of HCPs are really unique to the respective host and the manufacturing process. You won't see a certain level within the regulatory guidances of HCPs that are allowed within your product, although 100 parts per million is industry standard. ELISAs are actually are very simple to use in routine analysis, and they can quantify low levels of HCPs. So a lot of companies go to the ELISA, their high sample throughput. You can enable the process consistency monitoring. They're sufficient for a routine QC laboratory. They're quick to use. They're easy to train on. They're easy to validate. You can use generic HCP ELISA kits. They don't offer complete coverage, though, for all process-specific HCPs. Another thing about the off-the-shelf kits is that antibody changes require requalification, revalidation. You may also need to set your specs and go back and perform historical lot testing for each antibody change, just depending on how different it really is from the previous antibody used in the kit. And these commercial kits, they're generally acceptable for the early clinical studies, but they can be considered more inadequate by the time you reach phase 3 or commercialization, where regulatory agencies are often really encouraging process-specific HCP ELISAs. And I know that Eric's going to talk more about HCPs later on, so I'll let him get into more of the specifics. Another one is guidance on residual genomic DNA from host cells. I get a lot of questions from people on this. Residual cellular DNA is highlighted as a common impurity with a potential risk for biological activity. So it's really important to monitor and control it in your final product. From the FDA guidance on CMC information for human gene therapy investigational new drug applications, they talk about, and really this is pretty consistent across the industry, the size of the residual DNA should be reduced to below 200 base pairs, which is considered below the size of a functional gene. And the amount of DNA should be limited to 10 nanograms per dose, which using calculations that I talked about earlier, you should be able to figure out. But as also, when you're talking about residual genomic DNA, you should be really pay close mind to the transforming sequences that you might find in your HEK293 or other host cells. For example, in For example, in the HEK293 cells, the adenovirus type 5 early region 1A, this was used as a transforming sequence and is considered a sequence that could potentially be oncogenetic in your product. So you're going to want to specifically test for this within your within both your vector and probably your drug product if possible. Luckily, this transforming sequence is not a human-derived sequence, so it's much easier to test for residual level amounts of E1A in your drug product than it is for the rest of the host cell DNA. Okay, and just in summary, impurity is any component of a drug substance that is not a chemical entity defined as the drug substance, and for a drug product, any component that's not a formulation ingredient. So basically anything within your product that you don't want or that isn't necessarily causing the efficacy of your drug product. You'll divide those into product-related and process-related. We talked about process-related here. Control of impurities is important for many reasons, but really safety is our #1 reason, and you want to keep that in mind throughout any assessment of your drug product impurities. CQA assessment is based on the predicted severity and uncertainty and knowledge of the impact. Regulatory guidance is Q3C and Q3D. I highly recommend if you're interested in impurities that you go give those a read. They give very useful information for calculating your permissible daily exposure for impurities. And then of course, there are always several ways to think about meeting your PDE beyond testing the final product, including spiking studies, theoretical reduction, and risk assessment. We talked a little bit about those today. We will do Q&A at the very end after Eric's given his talk too. So with that, I will pass this back to Marcus and Eric.
That's great. Thank you very much for that, Jessica. Great presentation there to start us off in today's webinar. Thank you very much. And a quick reminder to our audience members, if you have any questions for Jessica or for Eric, then please make use of the questions tab located directly below your webinar screen to send in your questions. And we will go through these questions in our upcoming Q&A session after this next presentation. So now please allow me to introduce our next presenter. We have Eric Bishop, who is the Vice President for Research and Development at Cygnus Technologies. And Eric leads the Research and Development team and is responsible for new products development and custom development services. Eric has 20 years of experience in HCP analytics, and he joined Cygnus Technologies in 2010, bringing a background in R&D for several biopharmaceutical companies. Prior to joining Cygnus, Eric worked for AstraZeneca, formerly MedImmune, from 2002 to 2010. And during this time, he had multiple roles from HCP assay development and tech transfer to being an analytical representative on CMC teams, guiding projects from IND to BLA submissions, as well as head of new technology development group. And prior to AstraZeneca, he was at Crop Tech Development from 1998 to 2002, where he was involved in the characterization of recombinant protein produced in plants. And with that, I'd like to hand over to our next presenter. So welcome, Eric.
Thank you. Thank you for the great introduction, and Jessica had a great segue into this part of the talk for host cell proteins. So we're going to talk about analytics for process-related impurities in viral vector manufacturing. So just a little bit about Cygnus Technologies. For those of you that are not familiar with us, we have been in the process-related impurity testing since 1998, when we introduced our first CHO and E. coli host cell protein ELISA kit. We've worked through most of the early 2000s, just developing our portfolio so that we could have generic kits available for most of the expression platforms that were used across the industry. 2008 is when we first started doing some of the orthogonal services testing, again, to help understand the quality of the reagents we're using for our ELISAs. 2014, we developed and launched our antibody affinity extraction. That's a superior way of doing coverage analysis, so that, again, you can understand the quality of the reagents you're going to use. And a lot of the talk today is going to be revolving around that. 2016, we were finally able to merge mass spec into host cell protein analytics, and that's really paved the way for the talk today, but a lot of the great new things that we're doing in host cell proteins. And then finally, in 2020, we released our MockV portfolio of products. So we're using virus-like particles to basically mimic or to substitute in for viral clearance, so that you can do viral clearance studies on your bench rather than having to send them out to contract labs. Okay, so this is a relatively new field. Biopharmaceutical manufacturing has been around for a long time, and we've learned a lot with respect to host cell protein analytics from the monoclonal antibody field. So first and foremost, host cell proteins are they constitute a major process-related impurity. We know from experience now that they do play a role in patient safety issues, but also in product stability. We have a lot of bad players like lipases that can, when found in your product, can cause problems with excipients. So some of the things you may put in there to stabilize your product, and again, others such as proteases that can cause degradation or aggregation of your product. There are set limits right now for most of the regulatory agencies, but again, those are more based around what we've done historically with monoclonal antibodies. So I would suspect that will change as viral vector manufacturing becomes a little bit more prevalent. So here we have a list of a lot of the common expression platforms, the top left. These are a lot of the expression platforms used by viral vector manufacturing. We have generic kits to all of the common platforms. But also in process-related impurities, we're talking about removal of the host cell DNA, and also common media additives such as HSA, transferrin, some of these other things that we add into our culture media. And then finally, affinity ligands. So now there's more and more companies releasing affinity purification style for AAV, and those ligands tend to leach off the column. So if you're using those type of ligands and columns, you have to demonstrate that you're removing any leached ligand from your process as well. Okay, so first we're going to start with just the basics of host cell protein immunoassays. This is just more to get everybody on the same page so we can understand the rest of the talk. So host cell protein ELISAs are very complex tools where you're actually measuring hundreds to thousands of individual host cell proteins within a single well. So at Cygnus, we've taken it upon ourselves to make this very complex thing as simple as we possibly can. Because we know that companies around the world are using these kits, and there are analysts with different levels of expertise. And we want to make sure that everybody's successful as they can on the first try. So as such, most of our kits come as simultaneous incubations. So the plate comes coated and blocked with the capture antibody. Then there is a simultaneous incubation with the sample standards controls with the detection antibody. That's typically a 1 to 2 hour incubation, followed by a single wash, and then detection with TMB. So these can typically be done from start to finish in about 2.5 hours. So as such, again, the ELISA is the gold standard for process monitoring as well as product lot release. And it is for a couple of reasons. The most important is that it's the only tool that we have that can measure nanogram per mil levels of host cell protein in the presence of mg per mil levels of product. And as long as it has been expertly developed, it doesn't require a high level of expertise to run. So you can give it to entry-level lab technicians and they should be relatively successful. These assays are very reliable and robust, which makes it easy to transfer, whether it's down the hall to your QC lab or around the world to other CDMOs. Now, the ELISA does have some limitations, just as any method that we use for host cell proteins. And the first is that we're not actually measuring nanogram per mil of host cell protein. That's what we call it. It keeps it simple for everybody. But really, we're measuring host cell protein equivalents. And when I say that, what I mean is that, again, there's hundreds to thousands of host cell proteins in these wells or in these samples, and you can't actually measure each of them individually and get a total using these antibodies. So what we do is we have a host cell protein standard that we know the total concentration of all the host cell proteins, and we dilute those down to make the standards of the assay, so that the signal of each of those standards is, you get that, you can plot a standard curve, and then when you have an unknown, the sum total of all those signals in the well are measured against that curve, so that you can get an idea of total concentration. But you know nothing about the individual host cell proteins, and also that the results are subject to change if you change kit reagents. So if you immunize different groups of animals, even with the same antigen, they can respond differently to the different host cell proteins. So it's important to not change reagents, or to change reagents very rarely. And then finally, again, because we're making very broadly reactive host cell protein antibody pools to thousands of proteins, it's not a quick immunization procedure. So generally speaking, to develop a good host cell protein ELISA, start to finish to qualified assay, typically about 12 to 14 months. So we're going to talk a little bit here about the assays and what you need to do to use these assays from a regulatory perspective. For the purposes of this talk, we're going to not talk about dilutional linearity, accuracy, or precision. These are pretty well captured in a lot of the regulatory documents that already exist. So I really want to spend more time on coverage analysis and the characterization by AAE Mass Spec. That's certainly the newer things that are coming out, and certainly the most interesting at this time. So the first is coverage analysis, and we'll do a little bit of a history lesson for coverage analysis. When we first started doing host cell protein monitoring with ELISAs, we were really doing it as a process monitoring tool. In the early days, there wasn't a lot of thought given to the fact that host cell proteins could potentially be problematic. And we weren't doing this very long before we realized that wasn't true. So regulatory agencies realized that we're, again, measuring hundreds to thousands of proteins in these wells, and they started questioning the quality of the reagents. So they require a sponsor to prove or make them feel comfortable that if you are reporting low levels of host cell protein in your product, it's because you've done a spectacular job of clearing host cell protein from your product, and not because you're using a poor set of reagents that are insensitive to most host cell proteins. So that was in the early 2000, and the only tool that we had at that time available was 2D Western blot. So 2D Western blot is, you run the protein on two gels. One of them is silver stained. One of them you transfer to a PVDF membrane, and you probe that with antibodies. That was the tool we had, and it got us through for a while. We did it at Cygnus, but we also realized over the course of a couple of years that the whole point of the exercise of coverage is to demonstrate antibodies are broadly reactive to the host cell proteins in your samples. And we realized that that really wasn't the case with the Western blot. We didn't feel it was predictive. So we developed what we call the antibody affinity extraction as an alternative to 2D Western blot. And you can do that with 2D large format 2D silver stain detection, or LCMS. And that's what we're going to be spending quite a bit of time talking about today. So first, antibody affinity extraction. So this is a schematic of how this looks. We take the antibodies that we plan to use in the host cell protein ELISA, and we make a column. And we can do a lot of conditioning of this column. And when we're satisfied that it's ready, we take harvest material, so typically null or mock harvest material, and we set some of that aside. And we take the remainder of that material, we pass it over our column containing our host cell protein antibody. We wash the column to baseline, and then we do an elution. And typically we do 4 full cycles of this so we can make sure that we get enough host cell protein for downstream analytics. We take our 4 elution fractions, we pool them, we concentrate and buffer exchange. So at the end of this process, you have 2 samples. The first is the harvest material, which are all the host cell proteins that could potentially find their way through your process and into your product. The second is the AAE elution fraction, which are all the host cell proteins that the antibody was specifically able to pull from that sample. And then you take those and you have two choices for actually analysis. The first is just large format 2D gels with silver stain detection. You can use some nice imaging software and you can count the spots, overlay the gels, and then that gives you a very good assessment of coverage. I think much more recently, and I think much more valuable, is most companies are going over to AAE Mass Spec. So we take that same pre and post sample, but then we analyze each of them on mass spec so that we actually identify every host cell protein that's in our sample, as well as what we're reactive to. And we get some other information such as molecular weight, isoelectric point. So the first and main question that we ever received regarding our antibody affinity extraction is, we said we're doing it because we really don't like the sensitivity or specificity of a 2D Western blot. And we needed to demonstrate that the host cell proteins that we're removing from the sample with our AAE column is specifically removing those host cell proteins and not just nonspecific interactions on the column. So this is the experiment that we've set up to demonstrate this. This is using our F650S HEK antibody. We have our affinity column on the top, which is the antibodies used in the HEK host cell protein ELISA. We made the column, we loaded the sample. You see we washed to baseline, and we get some nice elution peaks for each of those. And as a control, we made a non-immune goat IgG of AAE column and ran the same harvest over. So you see the large load peaks and then washing to baseline. And again, very little to no elution peaks there. So we feel comfortable that when we see host cell protein in our AAE elution fractions, that they are specifically removed and that we can feel comfortable that the antibodies recognize those host cell proteins. Here's the typical workflow for our AAE with silver stain detection. So we have AAE fractionation followed by sample prep. We do horizontal gels, and then we have some nice imaging software that can take pictures of the gels and then do the full reduction of spot counting, overlay, and coverage assessment. The mass spec workflow, quite similar. So we have our fractionation by AAE, sample prep, injection on the mass spec, and then we have these very large data files that we have software to help interpret and identify the host cell proteins, help us assess coverage, make our virtual gels, and also help with report writing. So this is one of the things that was a limitation. There's a couple of limitations of having host cell protein analytics with mass spec. The first was trying to figure out how to see host cell proteins at such low levels in the presence of very high levels of product. We've worked through that. We'll talk about it in a minute. But the second one was not having databases available, so we can, you can do the mass spec, but if you can't interpret, the data, it was very challenging. So we've curated proteomic databases for pretty much all of the main expression platforms used for viral vector manufacturing as well as monoclonal antibodies. So here's some example data for our HEK antibody coverage analysis. Figure A on the top left is large format 2D silver stains. Again, so it's typical silver stain imaging. It's not that useful or meaningful until you have imaging software that can, again, identify, count the spots, and do the overlays. Figure B here is our virtual gel for an AAE mass spec. A lot of people, the list of identifications, doesn't really do it. A lot of companies still want to see these gels. But this is a way of having the virtual gel image in a much more meaningful way. So we can color coordinate all of these spots. So the green represent matches and reds are misses. So you can kind of get an idea of if there's any kind of pattern or some idea of what you're missing. But here in our AAE silver stain, we have about 1,690 spots in the pre and 1,600 post. So coverage about 95%. In our AAE Mass Spec, we've really upped our identifications to over 3,000 in the pre, about 2,800 posts, with a still coverage of about 91%. I do like to take this opportunity to just reiterate that, when we do coverage analysis, the point of the exercise is to demonstrate the antibodies are broadly reactive to the host cell proteins in your sample. So it's important to not get too caught up in the actual number and just really remember the point of the exercise. So, here we're over 90% regardless of the method. So we can feel comfortable that these antibodies will probably be a good fit for an ELISA to monitor our process. Here is the same. The first one was with our actual antigen used to make the standards for the assay. This is data from a customer sample. This is AAE silver stain. We had about 1,454 spots in the pre, 1,350 in the post. So again, our coverage assessment using the gels was around 92%. This is a 650S again, different customer sample. You can see it's about almost 1,800 spots in pre and close to 1,400 spots post. Here again, the coverage was 77%. So again, we're trying to demonstrate that the antibodies are broadly reactive, and still 77% is still considered to be an acceptable coverage number for this for an ELISA. Okay, we have another example with SF9. So we have about 1,585 spots for pre and about 1,214 post, which puts our coverage assessment, depending on how you do the calculation, somewhere between 64 and 78%. And just another with Vero. With Vero, we have about 1,500 spots in pre and 1,200 post for a coverage of 62 to 79%. And then here's that was all the gel-based methods. But I said most companies are moving now to more mass spec. You get a lot more information here. So we can look at our virtual gels. So you can see all the spots that are matched and all the spots that are missed, or proteins, I'm sorry. Here in the coverage, again, about 74%. The other thing that's important here is that all host cell proteins are not created equal. We know for sure that there's certainly some host cell proteins that are more likely to cause problems, again, whether it's drug stability or patient safety. So we also have the ability to know what host cell proteins we should expect to see in a Vero sample that are potentially bad actors, and we can make sure that the antibodies that we're making at least hit those. So you can feel comfortable, when you're doing your assay selection, that it's not just that it has very high coverage, but it's also hitting all the host cell proteins that we know that are going to be problematic or potentially problematic down the line. And here's a Vero with a customer sample. Again, the nice virtual image. Here our coverage has gone up a decent amount. We're at 88%. And again, you can see that of all the potentially bad host cell proteins that are there in the pre, that we're also detecting them in the post. So everything that's there, we're detecting. Again, it's just more data that you can use for yourself to feel comfortable, but also to present to regulatory authorities that the assay that you selected is appropriate for use for monitoring your process. Okay, now bridging host cell protein antibody pools. So again, when you change host cell protein reagents, we all know that you're going to see differences. And it's important to understand what those differences are as early as possible. And historically, we had no ability other than bridging studies in the ELISA. But now we can actually look at the quality of the reagents, the previous assay or previous reagents you were using versus a new one, to really get an assessment of if they're still appropriate. So here is a HEK. We resupplied this assay a couple of years ago. The new assay, we detected about 3,000 spots. Again, we had 2,800 in the post, so 91%. But again, on the list of potentially bad actor proteins on the right-hand side, you can see of all the ones that were there, we were reactive to them in the previous F650S, as well as in our new resupply assay of the 650S. So we can say with some level of confidence that the new reagents that we're using for this kit are at least as good as the previous, and we know that they can hit all the potential bad actors that we were able to see before. And then here's more of the head-to-head bridging. So we did a single antigen. We did the AAE Mass Spec for both of them at the same time on the different. Sorry, both of the resupply and the older version of 650S. Again, both of them are in the around 3,000 identifications and about 2,900 in the post. So coverage range of 90 to 96 before and 90 to 91% now. Again, both of them are well over 90%, and we can feel very comfortable that both of them are going to be appropriate for use in the ELISA. So I do want to take this again. So we talk a lot about coverage, and I know I've said it before, but coverage is just as dependent on the method that you use as it is on the quality of the reagents. So it's very important to not get too caught up on the actual coverage number, and really just more of being comfortable that the antibodies are broadly reactive. An example here, I don't think we have the data, but for the 2D Western blot, we have our same. This is a CHO example, I believe. But we run the same reagents, so the antibody and the antigen in 2D Western blot, AAE silver stain, and AAE Mass Spec. And you can see coverage differences, ranging from 50 to 90% on the same reagents, just using the different methods. So coverage is dependent on the method, and we strongly encourage you to use the best method that you can for coverage analysis. That's typically AAE with silver stain, but the best is AAE Mass Spec. It removes all the limitations of all the gel-based methods. So again, just hit again, coverage is dependent on the method. Just be careful of what you're going to use. Try to use the most modern methods that you can. It will help you in the long run. And then finally, the AAE is just much more predictive of how the antibody will perform in the ELISA. And just as a reminder, that is the sole purpose of doing this, is you want to predict how the antibodies work. Okay, and the final section here is the HCP identification and drug substance. That really is what we're all wanting to do and have been trying to work toward for a very long time, and we're finally getting to that point. This is kind of how it works. So we have our antibody affinity extraction column. So we have a brand new column that's never had host cell protein put over it. We pass our drug substance over for multiple cycles. We get multiple elution fractions that we pool and concentrate, buffer exchange. And again, gel work is not really an option when you're working with drug substance. So LCMS is really the only tool that you have to really identify these HCPs in the drug substance. So here's an example of actually following it through a purification process. So we can see here in step one, depending on how you calculate it, the coverage is somewhere between about 70 and 97%. Next step, 66 to 94. Third step, 66 to 94. And then finally, in the actual final drug substance, we identified 235 host cell proteins and a coverage of 80 to 89%. So it is a valuable tool to actually look into drug substance, and you can identify exactly what host cell proteins are making their way through and then finally into your final product. Here is a real-world example. Customer has sent us a sample to understand exactly what's in their final product. We're able to see about 264 host cell proteins in their pre, about 235 post. So it's 89% coverage, which is nice to know. So you know that of all the host cell proteins that are there, that you're detecting them. But just as importantly here is that you're identifying what host cell proteins are in your final product, and that allows for a more meaningful risk assessment. So right away, you can decide if there's proteins there that are going to be a problem with respect to your stability of your drug, but more importantly to the safety of your patients. So this is just kind of, an overall view of, the data that you get with mass spec as opposed to ELISAs or gel work. It's just much more informative as far as identification of what's there. You can test the quality of your reagents. I see that there's some concentration data on here before. I think it's still a little early in the game. We are absolutely not to the point where we can quantify individual host cell proteins with any level of certainty at this point. But that is certainly the goal of all of us that are working hard on mass spec, is to really find a way that we can more meaningfully quantitate individual host cell proteins. So again, we're not quite there, but that's where we're going. And finally, just to reiterate here, understanding the quality of the reagents and also the host cell proteins in your process are very important. We also know that most companies don't have this in their wheelhouse or the capabilities. So this is just to reiterate that, all of the things that we've talked about here are offered as services from Cygnus Technologies. And we also are happy to work with any customers as far as developing the process-specific assays as you move from early phase development into phase 3 and into licensure. Okay, so that's it for me. Thank you for your time, and I will turn it back over to Marcus.
That's great. Thank you very much for that, Eric, and also Jessica on the first half of this webinar of her presentation as well. So thanks a lot, and up next, we're going to begin our live Q&A session. So Eric and Jessica, I'm going to bring us back onto the camera on screen. Okay, one moment. Okay, very good. Thanks a lot. So our first question today will go to Jessica. Okay, and it says here, Jessica, how do you address the root-to-root extrapolation for PDE calculation, in brackets, which is relevant for parental application of CGT product?
Yeah, so that gets a little bit complicated. Within Q3D, they have a lot of examples where they split up their oral, parenteral, and inhalation exposures. But if you are looking in the literature and trying to calculate your own PDE, and you literally can only find an oral route, whereas you're obviously infusing into the patients for a gene therapy, what you can do is you can use the oral bioavailability as an additional factor on the denominator of your equation. I've seen this performed before. I don't have a specific example to show you, but using the calculated bioavailability will be really helpful in that instance.
Mm-hmm. Great, thanks a lot for that, Jessica. And before we move on, Eric, was there any additional comments to this answer you have?
Absolutely not. That is out of my wheelhouse.
Okay, thanks a lot. Then our next question, I think it's to Eric. It says, how much coverage is considered acceptable?
Oh, that's you will never hear a number out of a regulatory authority's mouth on that. It's all case by case, and rightfully so. There's some arbitrary lines in the sand that a lot of companies feel comfortable with 70 or 80% and above. But as far as from a regulatory perspective, it's case by case. It's the cell line that you're using. It's going to be the drug that you have, the frequency of dosing, and how it's administered. All of those play a role into what is going to be an acceptable coverage.
Mm-hmm. That's great. Thanks a lot for that, Eric. Jessica, is there anything you'd like to add to this answer?
No, I think Eric's the expert on the HCPs, but I would agree with what he said.
Yeah, okay. Thanks a lot then. Next question for Jessica says, is there a limit for E1A?
You know, in a perfect world, there would be, but the regulatory authorities tend to be a little bit cagey about what the actual limit for a lot of impurities are. Really, what you want to do is lean on your host cell DNA limit of 10 nanograms per dose, and obviously keeping the size under 200 base pairs as well. And then for E1A, I would just recommend doing a good characterization and testing and showing consistency from your process development all the way through your PPQ batches, that you're showing the same amount of E1A, preferably low levels, obviously, but there is no like one defined limit for E1A.
Okay, thanks a lot for that, Jessica. Eric, is there anything you'd like to add there?
There's not.
No? Okay, then. Our next question for Eric says, for HCP analysis in virus particles produced in HEK293 cells, do you determine only HCPs in the solution or include HCPs inside the viral particles?
Well, so I will say this is all fairly new to us as well, and we don't have a lot of feedback from our customers as how they're using these right now. So mostly in the solution.
Mm-hmm. Okay, thanks a lot for that, Eric. Any additional comments there, Jessica?
Yeah, I'd agree with what Eric said. In my experience, I've really only looked in the solution in the matrix of your drug substance or your drug product. But I know there are instances where people separate out their supernatant and their lysed cells in order to look at different impurities. I haven't necessarily seen that done with host cell proteins, but I wouldn't be surprised if people did.
Yeah. Okay, thanks a lot. Our next question for Jessica says, is the PDE listed in ICH-Q3C applicable for any administrational route, including IV inhalation? And do you suggest the use to use the PDE values in the ICH-Q3C without need for further comprehensive research and recalculation?
Unfortunately, the PDE values that they list within Q3C for specific reagents, they aren't super, I haven't found them to be super applicable to gene therapy impurities, which is why I really kind of stress determining your own PDEs. That being said, they do actually separate it out by the different administration routes within the guidance, which is super helpful if you are looking at specific impurities that are listed there. But yeah, like I said, I think that for the most part, for gene therapy products, you really have to do a lot of these calculations on your own. Should one of your reagents be found within the guidances, they do have it separated out by IV inhalation, oral routes. Do you suggest using the PDE values without need for further comprehensive research and recalculation? I mean, if they're listed in the guidance already, I'm generally pretty happy to accept that. If they tell me that 10 nanograms per dose of residual DNA, for example, not that that's more of a guidance than a PDE, but if they're telling me that, then I'm like, hey, that's awesome. I'm going to characterize my product. I'm going to show that we're below that, if possible, anyway. And I'm going to accept it and move on to my more difficult impurities.
Great, thanks a lot for that, Jessica.
Yeah.
And Eric, is there anything you'd like to add there?
Nope, sounds good.
Yeah, it does sound good. Thanks a lot then. Our next question for Eric says, normally the HCP level will be over standard limit for gene therapy products. How would you justify the upper limit of the HCP?
Well, and you're right, because the monoclonal antibody space has always had the advantage of affinity purification. So that's something we're going to be working through. Part of it's going to be manufacturing experience and early clinical data that will be used to justify higher limits.
Great, thanks a lot for that, Eric. And Jessica, anything you'd like to add there?
Yeah, I mean, I would just reiterate what Eric said, is that you want to look at your manufacturing history in your clinical trials. If you have shown consistently higher HCP levels, but you don't have any adverse events related to those HCP levels during your phase 1, phase 2, phase 3 of your clinical trial, then you can really kind of lean on that, and just process consistency too. If you're showing the same levels across, I believe within the guidances, and maybe don't quote me on this one, but I believe within the guidances, they don't actually say 100 parts per million is the limit for HCPs. That's more of like industry standard. Yeah. And what was I going to say? I believe what they say is that you really just need to reduce your HCP levels to appropriate levels or something very generic like that. So I think that you can state your case on this. That's not a solid limit.
Yeah, to follow up a little bit, yes, the 100 nanogram number came out of a publication from a long time ago, and it was just kind of adopted by the industry as, okay, that's the line in the sand. That is not in the guidance. And again, they will evaluate on a case by case.
Yeah.
That's great. Thanks a lot for your answers there. Okay, so our next question for Jessica says, do you have LRV values recommended for viral vectors?
I don't think that I can speak to everybody's manufacturing process. I will really just kind of harp on the consistency in your own process. I mean, you're just going to want to characterize what kind of LRV values you get for each of your impurities, and kind of show the consistency again from your early studies to your later like GMP and PPQ batches.
Mm-hmm. Thanks a lot for that, Jessica. And Eric, anything you'd like to add there?
No.
Okay. Our next question is for both, it says, but I'll ask it to Eric first. It says, thinking about an AAV generation process, have you noticed any particular upstream inputs, in brackets, DNA, reagent, media, enhancers, feeds, etc., that increase impurities like HCP and HCDNA?
Well, I think I want to defer this question to Jessica.
Oh, dang. I was going to defer to you.
Well, so again, a lot of the gene therapy type companies are, they're fairly new to the host cell protein part as well. And they're just starting to reach out with us. And we're just starting to talk about some of these things. Up until recently, a lot of companies knew they had to test host cell proteins, so they're buying some kits, but there wasn't a whole lot of deep thought. That they had so many other complications that they were working through that host cell protein didn't really make it to the top of the list. So we're really just now starting to work more closely.
Mm-hmm. And Jessica, anything you'd like to add there?
No, I wish I had more information about this, but I haven't spent much time digging into the process development and how the differences in the processes affect the host cell protein or host cell DNA. I've spent a lot more time on the analytics side of things.
Mm-hmm. I mean, what I can say from many, many years of the antibody space is that what we have found is additives or changing in reactor conditions and things of that nature, they tend to not really change the plus minus of host cell proteins, whether they're there or they're not there, but they typically regulate how much of each of them are there. So generally speaking, a lot of things like that don't impact unless they're causing more cellular stress, which expresses different host cell proteins.
That's great. Thanks a lot for your answers there. Our next question for Jessica says, where do you get the PDE of an impurity?
So some of the impurities, like I said, are available. PDEs are available within the guidances within Q3C and Q3D. There are exhaustive lists of different impurities. Those generally aren't the impurities we are dealing with too much within the gene therapy space. So you'll want to calculate your own. A lot of the times we've used toxicologists to actually perform these equations that I was showing within my talk, but the toxicologists will generally go to the literature and just see what's been done, what different studies have been performed, and what the side effects might have been for the impurities that we're looking at. And based off of that, you can use the PDE equation.
Mm-hmm.
Yep.
Thanks a lot for that, Jessica. And Eric, any additional comments there?
I do not.
No? Okay then. Thanks a lot. Our next question for Eric says, most of the adherent cell line for vector product have BSA. how would you suggest performing ELISA with Cygnus kit without BSA interference? Same question applies to Western blot.
Well, in the ELISA, it's not a problem. The presence of BSA isn't an issue at all. There was no BSA in the immunogen that were used to raise the antibodies, therefore there should be no problem. Western blot, again, I caution you about using Western blot in the first place. I don't think it's a very meaningful tool, but yes, it can cause problems there just for the fact that typically the BSA is at a relatively high level, and Western blots tend to get sticky. So you'll most likely detect BSA, even though the antibodies aren't specifically recognizing the BSA. So if, for coverage analysis in your samples, we would generally recommend doing AAE Mass Spec in that situation.
Thanks a lot for that, Eric. Jessica, any additional comments there?
I'd say if you're seeing interference in your ELISA, and obviously this is more Eric's area than mine, but ensuring that you're diluting out your interference within your product is really important to assess the appropriate dilutions, and that should be performed within the qualification of your method. Also, if you're using BSA in your adherent cell line for production of your vector, you're going to want to test for residual BSA in your products as well. Just shout out to BSA.
Absolutely. Yeah.
That's great. Thanks a lot for your answers there. Our next question for Jessica says, where can you find permissible daily exposure values?
Did I not answer that one yet? They're available both in Q3C, Q3D, and just calculating on your own using the information that's in my slide deck.
Mm-hmm. Thank you very much. Eric, any additional comments there?
No.
Okay. I think there's another question here for Jessica that says, how do you consider the fact that impurities can absorb onto cells for your safety assessment?
Well, I mean, when you're testing for different impurities, if you're worried about them absorbing to the cells, then I would recommend testing both your supernatant and your cells in your drug product, and separating it that way, and then reporting them out separately and showing. I think that's sufficient.
Mm-hmm. Yeah, that's great. Thanks a lot for that, Jessica. And Eric, any additional comments there?
I do not.
No? Okay. Well, we are coming up to the end of our time here. So thank you very much to all the audience members who sent in the really interesting questions. There's some great discussions going around these questions. And if you have any more questions for us and you're watching this webinar on demand, then please feel free to send in your questions to me and I will forward them to our presenters here today. So my email address is, I'll spell it for you, marcus.osei@biopharmawebinars.com. Okay, and just to remind you, you can watch this webinar and other webinars by going to our website, which is biopharmawebinars.com/webinars. Okay, and before I finish today's webinar, I'd just like to ask our presenters if they have any closing remarks. We can go in order of presentations today. So Jessica, over to you.
Just wanted to say thank you for listening to my talk, and thank you for the interesting questions. I've really enjoyed my time here, and thank you to Eric for a really informative talk as well. HCPs are a really big part of our business, and so it's always good to get an expert on the subject.
Thanks, Jessica. And Eric, over to you.
Yeah, again, thank you for everybody that took the time today to listen to our webinar. I also found it very informative. Listening to Jessica's talk. It's nice to hear the other side of this. And yeah, thank you for hosting. Appreciate it.
Mm-hmm. And I would also like to take this opportunity to thank our presenters today. So once again, that was Jessica Townsend, Associate Director of Analytical CMC Lead at Intellia Therapeutics, and also Eric S. Bishop, Vice President of Research and Development at Cygnus Technologies. So Jessica and Eric, thank you for both joining me here today. It was a pleasure to have you, and I look forward to working again in the future. Okay, so we're all done here, and have a great day.
Thank you.
Thanks, Marcus.
Bye-bye.
Bye.